Okay, so what I did was like I run library DEBrowser and I started DEBrowser with these two lines. And what I did was, so when you copy listening on this port and IP, it's a local host. It's my local host and this port. So I copied here and I opened two versions. See, one is DEBrowser here, one is here. So if you want to do two separate things at the same time and compare, you can always do those things. And also if you want to run, like you can always run it in your R here, another version with another from another port. Sometimes, I do that too, because sometimes your samples can be extremely complicated. You just want to open all of them and then compare by changing. So this is running in another session of the browser. And another session, if you couldn't install, for example, the browser, we have another version here, DEBrowser.umassmed.edu. You can go there. But this is slower than the version on my laptop. It's because using a server shared by everybody, and it's kind of make it slow. And also the other thing here is sometimes when your connection is not that good, or if there's a problem, when the connection is cut, then you're losing the session, and then you're losing everything you did. Basically, you cannot save it here. So it's like a browser. You have to load it and then do your things, and you cannot save the things here. But it's easy to do things. That's why we had this save option before, but I had to remove because of a couple of problems in the new versions. But in the future, I'll add this save functionality again. But let's do this now in the local version, which is faster. So I'm going to show you a couple of things that might make your life easier. So here, for example, there's a quick start guide, and you can define your own tables like this. So if you run these pipelines in Dolphin Next, you can actually download these files, and it can be top separated, comma separated, or whatever separation you use in your file. But in the Dolphin Next version, the file will look like this. So you have your gene names, you have your experiments, controls, or some samples in the columns, and the gene counts. And also we don't have it in Dolphin Next, but you can create this yourself, that you can define your metadata file. Like, this is, for example, treatments. And if you have batches, you can define your batches, and you can define as many columns as you want here, but make sure you're using exactly the same separation use. In this case, there's tab, and don't keep any spaces or tabs after the columns here, or like, if there's, and the sample names has to match with these sample names. And when the sample names start with numbers here, when you open the file, we talked this before, if it starts with numbers, it's put X before R. Or if you use a dash here, when you open the file, it puts dots rather than using dash. So R doing this kind of weird things. That's why I don't suggest people using like numbers in their naming. If they use numbers, just put X yourself here. If you use dash here, when you're defining your metadata file, put dots, but underscore is fine. And don't use other characters like, hash or something. Just use letters, numbers, and underscore. I usually do that, and I don't have any problems with that. But in that sense, R is weird. And also make sure that you don't have any string. Even you have one string or one character that is a mistake that you put here, or NAs that someone else has generated your table, and you have those kind of columns, and R will think that this column is like string, not numbers, and then you cannot use that in the browser, and you can get error again. Or if you put, but the count values can be should be numbers, has to be numbers. But here when you're defining your treatment, you can use strings, numbers, whatever you want. And when you're creating metadata file and upload metadata file to DEBrowser, it make your job much easier in many cases. for example, if you have lots of samples here, and if you want to make your sample selection easier, you can define 2x2 treatments, for example, or conditions. You can give any name here, by the way. You can give any name to any columns here. So, and you can give any, you can use any strings here or numbers while defining the conditions. For example, I just defined condition 1 and condition 2 here. And here batch 1, 2, just numbers, or you can use strings too here if you want to say like batch 1, batch 2, whatever. But imagine you have multiple more samples here for other conditions. You can define the condition 3, 4, but if you define those different conditions in different columns, and in one column, if you define only two conditions, and you can do the selection, and I'm going to show you in DEBrowser here, that when you load, so you have the treatment. So I just loaded as a demo, and I'll show you how I can use this treatment, because there are two conditions defined here. If you define the condition 3, you cannot use this column in the selection. So this might make your condition selection much faster when you do your DE, if you can actually define multiple columns, and each column you can keep empty for other samples, but put the condition numbers or strings to define those samples. So then your selection will be much easier. But let's do this, and how we are using DEBrowser and how it makes your life much easier. So we have filtering step to filter out all these zero count genes that Elisa was talking about in her talk, that I usually use max mean. These are without normalization of the counts, that you can remove those samples here that these are zero counts. And you can define your breaks if you have like deeper samples. You can define more breaks, like 1,000 or whatever, and to see the distribution better and more like you know, when I increase, you're going to see what's happening. See, so it's like this, but this tells me better that where it is exactly, and I use usually 100 breaks and then bin them in this, and you can see 1,000 of them. When you increase the bin, the number of genes are less in specific bins here, but zeros are there, 1,000 still, and then, but you can still see the separation around here. But more or less, you need to choose a number, and I'm just gonna this, and okay, so, and you can choose your max, and mean is the mean of the samples. And what it is doing basically is this. So when I press the show data and I see some genes here, for example, this is 0, and some genes see this gene, and maximum value in control is 7.97 when I check all the samples. So if I choose this max value 10, it will remove this gene as well. And this gene will go for, because it's all 0. And you want to, and also this gene will go away too, and this will go away too, that you don't want to keep them because the counts are extremely low. And also there's another option, that CPM. This is the CPM definition is somewhere here, data assessment. Yeah, it's row counts divided by library size by normalization factor. So this is actually normalizing by calculating some factors for each sample and multiply by like, you know, divide by 1,000,000. So this is count per million value. This is kind of normalized without using lengths. And what you can do with this CPM, when you're filtering, you can choose, like we usually use like 1 CPM, at least many number of samples. So it can be like 3 samples. So make sure that you are covering at least 3, 4 samples if you have 6 samples, because because if you don't cover many samples, then one sample can be like huge, like more than CPM 1, but the others can be all 0. So, and that can be on the like, you know, edge of like 1 or 1.1 or something, that CPM value. You don't want to keep them. That's why when you increase the number of samples, that might work, but you don't want to increase too much. That's a trick there, because, for example, if you are comparing control versus experiment, if you increase the number of samples more than 3 in this case, for example, in experiment, everything, all the gene counts can be 0. Then if you make this 4, then you might lose that gene contribution, even you want to take it, because maybe control, the gene counts in control are huge, but here they are all 0. So you need to make sure to choose this number wisely. Otherwise, you're going to lose those genes that might be valuable for you. Okay, so I'm going to use max and filter. So after filtering, see, you can see before and after. So these are gone. You could do a better job by increasing this value a little bit more, but it's okay. You know, these many genes are not going to change PHS value calculations too much, because most of them are here. And when you can look at the beginning, that we had 30,000 genes in this table, but now we have 40,000. So we eliminated 16,000 genes here. That's perfect. And this affects a lot. And after you do this filtering, so what I do usually, if you think that, or if you know that you have batch effect, or if you just want to check someone else's data, if they have a batch effect, you can just go to batch effect correction. And I'm just going to show how I'm doing. So this is calculating the variance. So this is kind of unbiased way of doing batch effect correction. I'm just going to color the fields with the batch. See, these are different batches, for example. This is just an example. This is not a real batches, but, and the shape with the treatment, for example. See, these two in condition 1, and the shapes are this triangle here, and the circle is condition 1 again, and the red color. So you can do these kind of things if you have multiple conditions, more than 2 conditions. You can put them together here and calculate the variance. This calculates the variance for you. And then you can see if there's a batch effect, like Elisa showed you how it is looking before batch effect correction and after batch effect correction. And you can also check the normalization and other plots. This is before normalization and batch effect correction and density plots. If your data is skewed in one way, if the counts are low, for example, in one sample, you can see them easily here and then remove. These are like kind of QC and batch effect detection step. So if you discovered a batch effect here, what you need to do is you need to define those batch effect, by the way, in the columns in your metadata and your treatments. And normalization is a must when you're doing batch effect correction. Elisa talked about all these normalization methods like TMM, RLE, and Apu similar methods coming from a jar package. mRNA is coming from DESeq2 package that Elisa talked about them. So I'm going to choose mRNA normalization method, and correction method is combat. So Elisa also talked about combat methods. And you need to choose your treatment and batch as the columns. Again, you can define any name here, any naming for the header of these columns, but you need to choose that column here when you're doing your batch effect correction. And sometimes some donors can be different, and you can use donor as a treatment, you know, like batch, because different donors might introduce some more batch problem. Even there is no batch effect, but donor has a different health conditions and stuff. You might need to use that donor in the batch just to correct the data and then do your experiments on the conditions like healthy versus, for example, cancer, whatever the condition is. Okay, so when I submit this, it will do the batch effect correction, but this is not going to do anything here because I know that this is the same data. There's no batch effect there. But still, when I do this, it's after normalization. This is just the normalization effect, by the way. After normalization, you can see I just change it to, like, you know, these are the colors are the condition 1 in this case, just change. And you can see before and after. So this is kind of good that you had this batch effect problem, and then after you correct, you have a better separation in PC. What you do usually is you can check this PC1 and 2 values. And if this number is high in PC1, it means the separation is good after batch effect correction in between your conditions. So this is what you're looking for to test if the batch effect correction worked right. But if this value is lower than your batch effect, lower than this separation, for example, there's no, but there's no batch effect, by the way. That's why you don't see it, but you can actually decide which one is better, applying the batch effect or not applying, because sometimes when you apply the batch effect, you might lose the real difference between the conditions. So it might affect your findings. So you should be sure that you have the batch effect by looking here, like Elisa showed. Like, for example, she was working on time points, and for per donor, the different time points actually got together in clustering, and it shouldn't be like that. You know, the time points coming from different donors should be clustered together, not all donor samples clustered together. Okay, so if that's the case, you need a batch effect correction for using the donors in your batch. And then after batch effect correction, actually we have that sample that Elisa showed in the demo section, that if you you know, come here, where is that? So this load demo donor, that's all. So if you load this one and play with the data, you can see the batch effect and correct it in a better way. Okay, so what I'm going to do now, after batch effect correction, you can either go to D analysis or do more QC plots. I'm going to go to QC plots to show you a couple of more things that you can play with your data to understand if anything is wrong. So for example, there are a couple of data options here that you have to be aware that you can change the data set. You can change all detected genes to all detected genes that I did now, means that 14,000 genes that after filtering, that you can play with that data. So for example, this is the PCA of that 14,000 genes. And the table is that 14,000 genes after normalization and batch effect correction in this case, because I used that data. And you can, so you see the same PCA plot after batch effect correction, and you can use the QC options here to use those columns and legends. You can put the legend on and off. You can put the text on and off here sometimes just to see controls, and you can download this as PNG, or there's another option here that you can click download SVG to download. These are support vector graphics that you can play with other software to, because some of the naming are, even the text is here, but it doesn't look good because R is automatically putting the location, and it's hard to change them here. That's why, you know, you need to play with these plots a little bit using other graphical interfaces. Another PCA options here, you can change the PC. So these are only PC1 and PC2. If you want to check other dimensions, other separations in the third or fourth PC dimensions, you can actually change this to the 3 and 4. Usually, you know, we don't do this too much unless the separation is not in the first 2 dimensions that cannot, and the separation cannot be explained in the bigger variance in your data, then you can go lower dimensions there in the PC results. But these are the PCA plots, and these are the all-to-all scatter plots that you can. By the way, one more thing I was going to tell you. In the PC plots, how you can decide which dimensions I should go more, when you see this PC1 is very low and you see higher PC values here, PC3, 4, and 5, then you might want to check those PCs. Why there is a huge variance in this dimension? What explains this? Some data can be extremely complicated and can create variance in different dimensions in the data that you might need to go and understand that variance here. When I say variance, it means the variation in between all these samples means, so for example, the variation can be in the condition or in all samples here. So it's just basically doing like unbiased way of variance calculation. And I will show you what the variance means in the box plots too. But basically the variation in the conditions and in all samples. Any questions there?
Yeah, how did you get the scatter plot?
I will show.
Oh, here on a plot type, sorry.
I'll show.
Okay.
Are you talking about this plot or?
The scatter, no, the scatter plot that compares each, you know, the.
I'll show in a second.
Okay.
So here, like these were the QC plots, and you check all told scatter plots here. So these are like the all told scatter plots using all detected genes. You see like 99.99 all detected genes, and experiment 99.98 very high. And when you use all detected genes, these are kind of lower. But if I change the data options to most varied genes, and the most varied genes are the genes are here, like uh, in all detected genes. The genes are more varied when the counts are lower here in this area, and less varied when the counts are higher. And usually the accuracy, replication accuracy higher here because you get more read counts when the gene is more abundant because of the sampling bias when you're doing sequencing. But when the gene has a lower abundance, for example, you get 10 reads from that gene, and in two different experiments, you can get like 5 or 10 in this area because there's a huge sampling when you sequence 50 million reads. Think about like 10 reads out of 50 million reads, and those 10 reads are in this area coming from that gene. It means extremely low abundance. And that's why replicability is low in this area, that when you calculate the variance using this data set is doing that actually. And if you keep total mean count low, like 10, you're actually exploring, and top 500 genes, you're exploring low abundant genes here. But when you increase the total mean count to, for example, 1,000, you're actually going to the middle part of your data. See, in the middle part of the data, without doing any differential expression analysis, by just looking varied genes, most varied genes, like top 500 most varied genes, when the minimum count is 1,000, I started seeing the separation. So this means that this data is beautiful. It's already showing me some differential expressed genes without doing differential expression analysis. So if your data is like this, you are lucky. And this data is uh, like Vernier et al., done here in UMass, and they did a beautiful job and clean job that without doing any DE, you can actually see the separation. And when you look at the biological replicas now, see 97, 97, even these are the most varied genes in between these two samples, still the correlation is extremely high and very replicable here, you see. And here, the correlations are lower. And even when you use total, like all detected genes after filtering, the correlations were 97, 96. Now it goes down because these are most varied genes. So you selected the genes you want to work on. And you can actually create this heat map by using this. But okay, this is unbiased way of finding the genes if your data is beautiful like this, but there's still some variation here you see in the heat map that some of the genes here, as you can see here or there, there's a huge variation. Maybe these are not real DE genes. Maybe the variation is huge, but there's still difference, but not replicable in all the treatment. That means that maybe you shouldn't use them. That's why we use the differential expression analysis to remove this variation bias. That sometimes there's a huge fold change in between, for these genes, there's a huge fold change between this and that when you look at only the average of these genes, but this is not repeatable in all the experiments you perform, all the treatment you perform, you should remove them. So differential expression analysis is doing that. So this is the QC that you're applying. If you see a huge problem in one of the sample, you decide removing that before you do your analysis, and then you move with your analysis after you remove all these problematic samples. And by looking this most varied genes, you're proving that your experiments work pretty well to yourself. And then you go to differential analysis, differential expression analysis. So it's here, there's the link. And when you do your differential expression analysis, you just add comparisons here, add new comparison. So in this case, I want to show you like condition 1, condition 2, and you can apply different methods. For example, I'm going to use DESeq2. These were row counts. After batch effect correction, actually, I did batch effect, but I just want to do with the row counts. Just refresh this. I'm going to load again. I'm just going to do my filtering with max. And without batch effect correction, I'm just going to go here. And so here this is select meta. So you can actually use this to do faster selection. That is what I was talking about when I was explaining metadata definition. So when you define different columns here for different sets, you can easily select without using these boxes. Otherwise, kind of makes your job slower when you use these boxes and remove, for example. In the second comparison, I want to compare 2 replica versus 3 replica. That I want to show you why you need, why more replicas helping you better while you're doing your analysis than 2 replicas. This will also tell you. But also you can change the DESeq method, HR or lemma. If you want to do, if you have, for example, microarray, microarrays are already normalized counts. So you cannot use DESeq2, we are using lemma, for example, for microarrays. Or if you want to play with the other parameters here, change test type, or for example, if you want to do GACA analysis to detect this Go term that are enriched in your data, you need to use shrinkage that calculates a statistical value using like wild statistics, for example, that if you choose, and it's sorting your genes using that statistical value, then that sorting is extremely important for GACA analysis, because it's going to tell you in that order of your genes, the top number of genes are enriched in this category, and it gives you a plot. So just make sure you're using shrinkage when you do your GACA analysis and order your genes. Kind of things make your life much easier. And then, so I'm going to start DE. So this will do 2 differential experiment analysis now using real data. And you can see, this is the first one. You remember when we were doing before here, we saw this these lines. And the second one started here, estimating dispersion. And why you're doing multiple comparisons, sometimes you just want to compare different conditions and combine them and check the results. So when it is done, just scroll up if you're down there and go to main plots. There are other things that you could do here, change your filtering, change your condition selection, you can rerun them. And this is just a page just to quickly look your DE results, how many genes you find in upregulated genes, how many genes you find in downregulated genes, and non-significant using these cutoffs. And it's the comparison 1 and 2. You can actually change the comparison. But when you go to main plots, actually, you are coming to the real area that you're going to start discovering your data. So the question before was like how you can actually see which gene was this. So when you hover, you see the gene name here. It's going to make your job much easier. You can see p-adjust value, log to fold changes, and then you can see the variation, biological variation. So in some genes, see, for example, this is not significant because one gene has 400 counts and the other's not. When I did my, in that plot that I showed you in the heat map, this one was one of them, that you cannot use this gene because when you look at here in the box plot, you see that it's varied a lot. And in the box plot, you see one gene is 400 and the difference, you know, if you just use the calculated, what was it? Fold change is 400, 304. See, fold change is huge. by just using fold change, you might make a mistake. So that's why you cannot use fold change. You need to use also p-adjusted value. That will actually give you a better estimate and also better finding. So in this case, another one here that is not significant because one count in one condition is 80 CPA one gene. So this is how you create these genes, how you hover them on and do this kind of analysis. So another thing you could do here is when you, for example, you can remove non-significant genes, the background. By the way, this background is not everything. I just put background data in main options, only 10%. But if you want to use this plot in your publication, just make this 100%. It's going to get slower because it puts just 100 points into the plot. And this will make a little bit slower, but I can do it for you. But it's just making heavier and using, it's not that pleasant. See, gray is more gray here, and it covered everything, all the gray points here. Otherwise, when I do 10%, I'm just randomly choosing 10% of the background and putting them to increase the efficiency or like, make it light. I'm going to go back now. So everything is interactive here. And you can change the labels here to make it shorter, longer, according to you. And you can search a gene, for example, just to write label search points. So there is a section here in the data options that you can search a gene, for example, FGF21 gene. And again, whatever you write is interactive and it's coming there. And searching the gene, and see, it's just every letter is even like working, and it's coming there. You can copy and paste. See, FGF21 gene is here. That this was the PHS value is low and very replicable. Sorry, you can click on this just to keep it on FGF21. Oops, why couldn't click? Okay, so this is FGF21. And you can see the box plot here. It's beautiful. So other thing you could do here, you can remove, for example, this genes, non-significant or whatever, and you can select any area you want to draw, for example, just to create a heat map for that portion if you want, like this. And you can, I'm just gonna, for example, I'm just gonna choose FGF genes and create like you can also copy and paste the genes you're looking for into this box. So the FGF genes are here. It doesn't look good when you put the labels, but you can remove that. So, but that green points are my FGF genes that I just searched. And you can put your gene list if you want to create a heat map. So what I did was I removed all the other layers in my plot and create my heat map just for FGF genes here. And also what you can do, you can make this heat map interactive. So this can be also interactive. When you hover these, they can also be here. They're all biological variation and box plot and bar plot. And this is also interactive. You can zoom in, zoom out if you have more genes here. And when you double click, it's going to go back to all genes. So these are selected genes. So in this data set, choose data set, there are many data sets. You have to be sure which data set you're working on. In this case, for example, up and down genes, you can just select up genes, down regulated genes, all detected genes. I'm going to show you comparisons. Most varied genes and selected genes. So this selected genes, actually the genes that I selected now, that you can use those selected genes in other plots. Like even you can do PCA plots with the selected genes, or when you, oops, I was going to go to QC plots. There's still QC plots here that you can just to use that selected genes to do your PCA plot. These are only those FGF genes, for example, I did my PCA, see FGF. And when you go to the tables, you can see all the information about these FGF genes. And if you just want to create that, play more with the heat map, you can click here or you can just create this all told scatter plus C. These are FGF genes. So you can use a group of genes to look all these things together, and everything is, again, I'm going to go to up and down regulated genes now. So these were up and down regulated genes, and red and blue ones, and the counts are normalized. And these are some tricks you could use. But if you want to play more by changing your PA-adjust value, you want, for example, more significant genes that you want to eliminate any chance that you can increase PA-adjusted value here, it will change the number of selected genes. Okay, so maybe I can also change this 4, that fold change 4 means log2 fold change 2. So same thing, it's gonna do the same thing that we did in the class. See, these are those genes, and but the PHS value is 001. See, these are very significant, and it means very replicable. So when you click, and the numbers are high, huge. 80,000 read is coming from this, on average, 80K reads are coming from this gene, and around this area, and this makes the p-value at p-adjust value low. So these are not significant there on top, but the numbers are huge, 150K. You can see the variation here is huge. Okay, this is what I was talking about, the variation. When you calculate the variance, if this is varied a lot, that variance number is higher. So this is what I was talking about that. And there are many other options here, but I want you to focus on, like, learn, especially searching genes you're looking for in this plot, and multiple genes, or using a part of a gene if you want to use, and the plot types. For example, some people like using volcano plots. It's the same thing, but another visualization method that you can actually use this. And sometimes some values are huge, like, some PHS values are huge, and all data goes skew here, and you can change a cutoff here just to put a, just to see the difference in this area better. So you can change this cutoff, and if it is more than 16, 18, for example, they're all here. So it means they are very, very significant. So this was minus log 10 PA-adjusted value. And this is also interactive. You can look at the genes, but it's a different way of showing the same thing. There's another one, MA plot. It's just multiplying here, like this is minus this addition in log scale. So in this scale, it's multiplying the read counts in normalized, because these are normalized counts. By the way, when we are visualizing anything here, when we are doing DE, they are not normalized. We get the DE genes, but we are using normalization here. In normalization method is MRN here, or you can change them. And when you go to table section, you can actually see those normalization values and data options up and down regulated. So you can download data and use these genes, like 115 genes, up and down regulated genes in other plots, other programs. And the second one is here. So these were like 3 replicas. And the second comparison here, this is, I choose a new comparison here, second one, and I only have control replica 1 and 2 here. With the same cutoffs, I have 229 entries, so it's more than 3, because in one replica, maybe it's not replicated in that one. So you can actually see this. But the good thing, when you want to compare different comparisons, we have this comparisons data set here in data options. And it's going to put them together, the fold chains in the first 2 comparisons and second 2 comparisons. See, this gene, for example, C7 something or GLG1, looks like here, it looks like significant when I use 4-fold change, because 0.22 is less than 0.25. And here it's more than 0.25, and still significant, very replicable when you look at the normalized counts. but you know, when you have 3 replicas, it's not in that cutoff that you are using. Or sometimes some of them are not even significant. In this case, my filtering is kind of low, but I'm gonna make this normal, like, you know, in the default, like defaults. So I'll show you more examples here that that you can use these comparisons, and some of them are here. For example, this case, when you have 3 replicas, uh, it is significant. When you have 2 replicas, it is not. So you can see it. So, and there are many things here, like these are not significant. Here there are significant. See, but these numbers are low, but still it's 0 in one, not in the other one. So these kind of things are very helpful. And also what you can do, you can download this data, the whole table, with all these calculations, and use them in Venn diagrams, for example. With this could be different conditions, by the way, not only in like 2 versus 3 comparisons, but this condition versus that condition, and this condition versus another condition. Then you can find the overlap in this table and then create some Venn diagrams if they overlap in some points. So, and the last thing I want to show you, I want to go to up and down regulated genes, and I want to show you some pathway analysis. So in the Go terms, so we have, if you come to this tab, you can apply some Go terms. So you can change the data sets. You can look at only up or down or up down together. I usually do up down, unless I'm doing GSEA. And then you can use like filtering options and stuff, and then plot type. So I'm going to show you, for example, KEGG pathway analysis, or you can do disease analysis, and you can change the other options here. We are using mouse, for example. This data set is mouse. You can change the organism, and this p-adjust value is different than the one here. This is the p-adjusted value when it calculates if a group of genes belong to a category, and it calculates the probability if that group of genes belong to that category by chance. So this is that value. If it is less than this value, its likelihood is less than this value that it belongs to that category, that group of genes. And then you can actually use this as an evidence that your data worked, or there are some interesting things in those categories. But what are those categories? Like, you know, you can just submit this and get the results. But these are not like, this is in R. In differential, these kind of things are just going to give you some preliminary analysis. And for example, this is the name of the paper, actual PPR signaling pathway was the name of the paper published. You know, this naming is inside of the paper title. So, and it's there, it's telling that this data worked, and you can actually come here and click of the description of the pathway. And then when you press this button, you can create the pathway. But again, there are better methods than how we are doing the analysis in R here. You can actually use other programs like David or Funk Associate, or they do like different kind of analysis that I was comparing them. But this is using the packages in R. Sometimes it's not perfect, sometimes it's limited, but still, you know, some of them are working like this. If it works, use it. But don't stop here when you're doing your Go term analysis. Go and use other third-party tools. Download the gene sets, background sets. Don't forget to download the background set and use them together. Okay, so this is another thing. If you are curious about which genes actually belong to that category, you can click here and see the counts. And these were 17 genes in your data set actually here, and the values are here after normalization. And the other thing here that I was going to show you, that these colors here on this plot, KEGG pathway plot, is actually your gene quantification results in your table. So it colors, these are, for example, up regulated, these are down regulated. See, upregulated and downregulated. And it tells you the situation here that you can use this in your, and download these plots, download plots. And there's another plot here, dot plot, that you could use. And you can change the p-adjust value to see more, if there are more, to see, more things here. And the table is like bigger, that all results are here. And also when you're using GCA analysis and stuff in other places, you can actually go to tables and download them. And let me go to, for example, all detected genes are important. When you are actually doing these are interactive. So like, when you change the data set, it's going to actually run everything you run before and then update those plots. These are all detected genes. And in the table section, you can download these results. Okay, I think I'm done with the DEBrowser. Now we have 20 minutes. I had another video, but I'm not sure if we have time. But what I'm going to do now is maybe I'll start, and if you want to continue watching, it was actually maybe 25 minutes. But maybe you can watch that in your own time. I'll put it into our things. Maybe I can answer your questions now about DEBrowser, that to use this 20 minutes like that. I think it's going to be better. And you're going to watch that Elisa's talk in your own time later. And also we have all this thing I want you to go over in your own time. Like we are telling how to use this in session 7. And we have lots of explanations here about how to use the program. And at the end of this, there is a homework. And I want you to do this homework too. And you're just gonna actually start using the program. And you will find the FGF21 gene, and you're gonna do the PCA plot, and you're gonna change the p-adjust value. And also there's something here. You're gonna compare DE62, HR, and LIMA results and create a Venn diagram using this tool that I found on the internet. But you can do this yourself in R2. It's up to you. And also, I want you to do, after you find the genes, you can go to David to do some go-to analysis using third-party tool David. Okay, and you can learn these things and how you can interact with other things. So what you need to do to do this homework after you run the browser, what you're gonna go is you're gonna come here after running the tool. So you need to download the data, all detected genes. And with Excel, you can do your filtering yourself. And you can download 2 files. One can be all detected genes like this, or the other one can be only up and down regulated genes. When you select this, when you press this button, it will download up and down regulated genes only. So then you can actually upload only the gene names to David and do your own go-to analysis using a third-party software. Okay, now I'm ready to take your questions.
One quick question is the discover option that comes. How do I access that one?
Which?
You had the discover option. Oh, is it?
Which one? Which option here?
Data prep. You have the tabs data prep at the top, right underneath the discover option. I don't see it in my discover. No, in the options. It's just the tabs data prep, and then next to it discover.
Discover is bringing here the main plots. It's the same thing.
Oh, okay.
After you run it, you're gonna see this, by the way. Not there yet. After you run, this appears.
Okay.
This is another way of going here, actually. See, when I click here, you discover. So this is the preparation step, filtering, condition selection, the analysis. This is the discovery things that you plot all these things. Some people are missing this. That's why I put it here. They forget to press this button. You need to press the Go Main Plots button that all these things are appearing after that. The other thing, maybe I can tell you a little bit about this. So when you create your heat maps, so for example, I'm going to create the heat maps here. This was scatter plot, but these are four samples because I selected, I think in the filtering, I put it here, choose comparison. So it's only the second one here. So let me do the first one. So it's going to create for the first one, up and down regulated genes. See, when you look at the old tool, up and down regulated genes are here now, how they are looking in the old tool plot. But when I do the heat map, so I'm going to play with this heat map a little bit. Maybe I can make it smaller a little bit. I'm going to change the font change to 10. It's going to be smaller heat map that when I'm playing, maybe you're going to enjoy more faster. But you can make this interactive. Interactive heat map is a little bit slower. You can define a title, my heat map, and you can change the sample labels. Like you can put a label here and see my heat map is under there. So you can change the size of the layout. Where is my scale options? No, where did I put that? Heat map size and margins. So plot size, I'm gonna change the plot size a little bit just to see these numbers better and the names better. And the margins, I'm gonna change the top margin to, for example, 69, just to see the name. And also usually I use the red upregulated, green downregulated. So there are heat map colors here that you can define your own colors by changing these numbers. See, this is, and you use custom colors, this is the default, that red is upregulated, blue is downregulated, but also you can go back and change some pre-built colors, for example, red and blue, like this, or blue green. I don't know, some people like other colors, heat, whatever. So it's up to you how you want to use or which colors you choose. Okay, this is how I use usually, blue, red. And the other thing here, sometimes you want to remove some of the things here. For example, dendrograms here. Sometimes you don't want to put this dendrogram to the side for the genes, and you can choose only columns, for example, and only columns. But you lost the, if you do this, like sometimes you give the order and it draws like that, and you can use another clustering here, like k-means, for example, to do the same thing. See, I remove that and I put k-means clustering. So it's another clustering sometimes you use. And you can increase the number of clusters if you have like multiple conditions, multiple, for example, time points. And if you want to visualize them and play with your clusters, rather than just doing DE, for example, this is not only for DE, these are in QC plots. You can upload your data here and play with the heat maps here with the number of genes you selected. You can also put the genes into data options here, the number of the genes that you are looking for, and play with your heat map here with these options that I'm using this a lot. With even without doing DE, I just use these options here. So I'm just gonna put that back, and I was going to show you, where's my dendrogram here? Okay, I'm gonna do the both, and I will not going to do k-means, and I will change, for example, column labels. I don't want to put the column labels, for example. You can just do this. Sometimes you want to put column labels later yourself. And, or you want to change the methods that you're using that I was talking about, like Euclidean, correlation, maximum Manhattan, Cambry, these things. Like, for example, correlation might work better sometimes. See, this is the correlation. And it's changing this dendrogram heights and stuff here. And if you think here in the column, you have multiple clusters in your samples. In this case, there are 2, we know it, but you can change the number of clusters. It will change the colors in the dendrogram. So, but in the row, there can be like 1 cluster here. You see there's a separation, another one here, another one here, another one here. So you might think like 1, 2, 3, maybe 4 clusters. You can change the clusters to 4, and it can separate them in the class. See, this is 1 cluster, second one, third, and fourth. Sometimes you use these kind of things in your plots. And I was going to do the correlation again. It's going to change the order, and it's much closer. Correlation works better. You know that. And there are other things like scale options that I was talking about. Like, you know, if I don't do the scale that I did with R, you can do these kind of things. Scale is not there now. See, scale affects the rows, as you can tell. There are like huge differences between rows, and they affect each other. When you scale, this dissolves that, like these things much better. And when you don't use center, you're gonna see like there's a huge white color here. It's not centered, it's centered. And when you don't use log, of course, the outliers will be affected. See, you see these kind of things. So these are things. Any questions about here?
So I guess about like the scale options, is there a time where you want to like get rid of the center or the log or stuff like that?
So sometimes what I do these kind of operations myself with it. Like, for example, I calculate z-scores in my data because of a reason. Z-scores has this kind of scaling and centering functionality already. So if your values are that kind of normalized z-scores, for example, it's kind of normalization. If you just want to use this heat map, you upload the data. Don't do DEC or any other thing, but use the z-scores in this table. And then in that case, you're not using scale and center or log. You're just using z-scores. That's why I did everything like parametric that you can actually change. And the pseudo count, for example, sometimes 0, 1, if your counts in between 0 to 1, for example, if you add 0, 1, it will affect the data a lot. Then you are increasing this value, decreasing that value. But as you can tell, it changes your plot. So you have to be careful what pseudo value you're using here. Okay, good question. And then if you think that some columns, some samples are not good, you can remove them. Like when you click, it's not there anymore in your plot. Another thing that you could do was like, there's a download here. You can zoom in some part of this plot. You could do those kind of things. And there's one more thing here. Yeah, heat map layout. So sometimes, you know, you want to change the angles and stuff. Like, you know, you can play with that like this. Or sometimes what I do, I make it like I have many heat maps, for example. I have only 6 samples, and I don't need to make this big, all the width. So these are like some suggestions. Where did I put that? I was, okay, heat map size and margins. So you can actually change. There's download SVG, by the way. When you click, you can download SVG from here, by the way, by clicking here. And what you can do, you can make the width like kind of like this. And sometimes you put them together, many of them, and using a single like figure that I use a lot. And sometimes I don't like these things, I remove them. So, but they have functionalities, these dendrograms, like when you use correlation, complete, but you can still, I usually use the correlation and complete, but according to your data, you can try different linkages, different methods, and you can read what linkages and There are good explanations in YouTube about like clustering linkage that you can watch if you want. Any other questions?
Hi, Honor. I have a stupid question. Could we change the order of the column name at the bottom? It says it's control 2, 1, 3. Could we change it to 1, 2, 3?
Are you talking about these names? Are you talking about sample names?
Yes.
Okay, so the thing is, like, you cannot change it inside, but what you can do, we uploaded this data, and you can actually, like, where is that? When you upload the data, you already have the data. Open the Excel sheet, change the naming, and save with the new naming, and those naming will appear here. Whatever the name in the file will be here. So you can change before uploading the data. You cannot change after that.
Okay. I mean the orders. You see the heat map. The order is replication 1, 2, 1, 3.
No, you cannot change again, the same thing. These data is coming from the data set, and you cannot change this naming inside of the browser. So you have to upload and do the same thing again and come here, and the names will be changed. But the thing, you can only add names, for example, sample label here with, for example, my samples. It's gonna add something here, and genes, for example, it's gonna add another name here if you need to do these things.
I don't want to change the name. I just want to change the order. The orders, you see, it's a control replicate 2, control replicate 1, control replicate 3. Could we change
The order. So yeah, that's the thing. So what you need to do, if you upload the file with the order you want, and you need to remove this dendrogram option here for that order you want to use. For example, if I just choose this as a row, the order will be, see, order changed. See, it didn't apply my clustering using the samples, and the original order came here. However, I uploaded control 1, 2, 3 here, experiment 1, 2, 3. Actually, I'm doing this sometimes. Sometimes I have one control and 1 hour, like, in time point, another same time point in the donor, for example. I put them together like this, 1, 1, 1, and I don't do this clustering in the row. I'm just doing the clustering only in the rows. But sometimes you do this, you order the rows yourself outside and upload that like that, and then you can just remove none, and this will be the order of the genes in your data set. So you can do that. But again, you have to order them before you upload the data. You cannot change the order. I don't have that kind of functionality to use drag and drop and stuff to change the order here. But good questions that maybe in the future we will have more functionalities, and we can think about it, how we can actually change the order. And
I have a question about fold change. So for some genes, for example, the value is 0 for controls. For whatever reason, let's say the gene was not expressed in the controls, and then in the experimental samples, it was say 9. There was a gene like that there. How do you calculate fold change? So when you calculate fold change, you divide the mean of the experimental samples by the mean of the controls, right? So if the mean is 0 for the control, how can you divide 9 by 0?
So the seq2 is always adding a pseudo count by looking the general distribution of the counts and calculating that pseudo count first and adding that pseudo count before getting the fold change. So I'm not calculating myself. When you run the EC2, it calculates and give you the log to fold change value, and then I trust that more than adding a pseudo count myself, because when you add the pseudo count yourself, like 0.1, while calculating fold change, you might make a mistake, because, for example, if it is 0 here and the read counts are 10 here, when you add a 0 count like 0.1, it's going to be like 0.1 in one average, like one condition, and the other going to be 10.1. And when you divide, it's going to be like 100-fold change. So you don't want to do that. So DESeq2 handles that for you. And then I'm using DESeq2 fold changes. And Limo is doing and Edge are doing that too. So they all have their own fold change calculations. If you're curious about how it is doing that, you can look at their paper about the fold change calculations. But yeah, this is a very big issue that you have to be careful, especially when the counts are low, that they might like, for example, you have only 10 reads coming from a gene and the other is 0. So when you divide without adding anything, then the fold change will look like 10 fold change. Sorry, infinite fold change, 10 divided by 0. But even you add 1, if the read count is 10 and the other is 0, when you add 1, 11 divided by 1 will be again 10 fold change. But again, like when you go down in the list, that is not a good thing. That's why when you use shrinkage, when you're doing this differential expression analysis, in that here, when you select that methods here, APJLM, for example, this is applying a method here. And it is when you do the differential expression analysis there, so it is calculating a statistical value, like I just removed this, and that statistical value is coming to the list here that I'll show you. And in that list, the order is taken into account the abundance value of the genes. And that's why those reads like, 13, these kind of things, where when the counts are low, the calculated
like, yeah,
stat
AK gene, for example, you see AK gene has 0, 0, 0, the third one. Yeah, AKA, AK2.
Yes, look,
yeah. So is there some sort of statistical analysis that happens in the background that we are not aware of that changes this 0 to some other value so that we can divide by 0? Well, by the new value.
It is changing it, and see, it found like a fold change 50, for example, for this. It just did it by itself.
Yeah.
Fold changes this. But the thing is, this might not be right because these values are extremely low and very varied. So this might not even be right, even DESeq2 is doing, but this stat value is lower when these values are lower. The stat value is lower. When the values are higher, the stat value will be higher. So when you sort with stat, see, and you can see better, like the genes that can explain better this difference. It means like, these is 0. There are some values here, like higher values, and the stat value is much higher. So this is what I was talking about. So in the other case, when you have lower values here, the stat value was like 16. It's down there in the list. Even they're differentially expressed, even they are significant, and even you can make these more stringent, by the way. I'm not doing the stringent here. You can change the method here to a wall test, for example. This is going to be more stringent and it will give you better results. So you have to play with your data. Look at that low counts. If those fold change is higher than you expected, then you can use another cutoff to remove them, or use the Watt statistics and use the shrinkage method, sort them, and remove them yourself. So something, I get an error for a reason. I don't know what happened. But yeah, if you get an error, refresh it and do the same thing again. And this, when you are running, that happens like, gray screen comes when you are using this version, if the connection is gone. By the way, we're done today, I guess. It's already 12. So I'm going to put one more video, like Elisa's video about probability, into today's session, into GitHub today. And when you have time, you can just go over this, what we showed today, and watch that video too. It's going to give you more insight about pathway analysis and like the probability calculations in those pathway analysis, p-adjust value calculations, and it gives you more idea about those things. And next week, we're going to do single cell RNA-seq. Arthur and Jake will do those things next week. that that you might have other questions about single cell, and also we can wrap up some of your more questions after we are done. So yeah, we're done, but I can be here to answer more questions if you have.
Guys, don't forget to try to install the packages for next week.
Yes.
As Alpi said in the beginning, these packages are there. There are instructions to how to install the package for the single cell data analysis that we're going to do next week. So don't forget trying that.
Yes. Yeah, if you get any error somewhere, just let us know. We fix those things that I got error. There's something like, you know, I see this X in selecting method for function something. So send these kind of errors to us that we are fixing.
Yeah, we get those errors all the time as well. So it's a bummer. So I mean, there's no way to do it.
There is order by X. It says something like this. So yeah, sometimes they change things in R that we don't know that they changed. And they affect our packages too, that we are discovering them from other users, and then we are fixing our side too. This is like a very collaborated world, but sometimes it makes our job harder if you cannot communicate with the community well, or if they don't communicate with the community with their own code. This happens to us. Any other questions? If you don't have, I'll stop it here, and you can continue asking your questions on Slack.